Okay, so today we're going to talk about inheritance. So we have talked about chromosomes, how they're just big molecules of DNA that's wound up. And we've talked about homologous pairs of chromosomes. And remember, homologous chromosomes have the same set of genes, but they might have different alleles. So if on this chromosome 1, I have genes A, B, and C. On its homologous pair, also chromosome 1, it's also going to have gene A in the same place, gene B in the same place, gene C in the same place, but the alleles might be different. And again, the alleles just differ in one or two bases, and that occurs via mutation, right? So we inherit one set of chromosomes from our parents, right? That gets like one set of their alleles. So mom and dad both make haploid cells, the sperm and egg, and then they fuse together to make the diploid egg, which is going to be you. Again, diploid means you have two copies of each chromosome, and haploid means you only have one. So each parent will donate one allele of each gene to their children, right? So we've been talking about alleles, and again, you know that an allele is just a different version of the same gene. It might vary in some, just one or two bases, and that humans are diploid, so by default, we all have two copies of each gene. And so in early genetics, people didn't know the first thing about DNA structures. They didn't know anything about A's, G's, T's, and C's. They were just sort of following traits. And so when they were doing this early research, they represented alleles by single letters that were either capital or lowercase. And so we'll talk about what that means in a little while. And again, for each gene in an organism, we'll have two copies for all of those diploid organisms, right? So there are a few different states that you can be in, either heterozygous or homozygous. So you have two versions of each chromosome, and on each of these chromosomes, you're going to have some kind of allele. If you're homozygous, it means that the alleles are the same. And so we'll represent these by like uppercase letters. So in this case, this is a homozygous genotype because it has two uppercase B's, and over here this is still two copies of the B gene, but it's just a different allele. So this is going to be homozygous little b little b. So homozygous means that they're matching as far as uppercase, and heterozygous means you have a mix of alleles. So these two in the center would be considered heterozygous because I have one version of a gene where it is represented by the uppercase B and another version of the gene where it is the lowercase b, so this is heterozygous. So in this case, we're pretending that there are only two versions of every gene. It's not quite like this, but for our purposes, for this next little while, we're going to pretend there's only two versions of a gene, an uppercase one, and then you have a different allele that's a lowercase one. And again, when the alleles are matching, when they're both uppercase or both lowercase, that's homozygous. Homo means like similar or the same, and heterozygous, hetero usually means different, so to be heterozygous means you have two different alleles of that same gene. So let's go ahead and do some practice here about whether or not something is heterozygous or homozygous. So in this case, the big A, those are matching, so that would be homozygous. For this example here, big B, little b, that's a mixed allele, so that's going to be heterozygous. And then for this one, we have an uppercase C and a lowercase c, so that will be heterozygous. So this is saying that it's got two different alleles of that gene C. And then like this example here, we've got two uppercase D alleles, so these are matching, so that is homozygous. So that is heterozygous versus homozygous. So now let's talk for a minute about genotype versus phenotype. So the traits that you can see. So this is the observable physical characteristics. So things like your hair color, your height. On this monster, it has a blue color, it's got eyes, one eye, it's got four arms. That would be a phenotype because it's a physical characteristic that we can see. The genotype would be the combination of alleles that gave rise to these phenotypes. So you'll, again, because the diploid organisms always have two versions of a gene, you'll see this as a combination of letters. So like AA or BB, in this case, these are two heterozygous genotypes. So genotype is going to be the letters. That's the combination of alleles, and the phenotype is an observable characteristic that you can see. So we used to call them traits. So not all alleles are created equal. So you probably noticed that sometimes they're uppercase and sometimes they're lowercase. And what this has to do with is whether or not the phenotype that results from that particular allele is dominant or not. So a dominant allele is always going to be represented by a capital letter. And for an allele to be dominant, what that means is that it only takes one copy of that allele to express the associated phenotype. So in this case, the genotype is uppercase A, and the phenotype is the color, which is purple. So in this case, the genotype is uppercase A, lowercase a, and the phenotype is still purple. So when we had sort of this fight between the big and the little allele, the dominant one, the big one, won out. So if the dominant allele gives purple, you only need one version of that allele to get the dominant phenotype. So you might remember way back when I talked about how mutations typically break a gene. So usually dominant phenotypes are considered like a working copy where you have a normal protein that is produced. So in this case, the uppercase A might be producing a purple pigment. So as long as there's at least one copy, the flower is still able to make a purple pigment because it still has that protein product that's allowing it to do that. The recessive alleles, on the other hand, can be covered up by the dominant alleles. So the recessive alleles are the lowercase letters, and you will only see a recessive phenotype when there are two copies of that lowercase letter. And the reason for that is that these are usually the mutated version of the gene, and they are usually broken and non-functional, right? So remember that mutations in the gene will give you mutations in your RNA, which will give you mutations in your protein. And usually those proteins aren't functioning anymore. They unfold, they don't work. And so when you have two broken copies, you can't make any pigment at all. And so you only see these recessive alleles when there are no working copies in existence, which is why for a lot of the heterozygotes, you will see the dominant phenotype when there's a mixture of alleles, because I have at least one working copy. So you'll see this theme over and over again, where the dominant allele has a working copy present, and you usually only need one to work before you have a working phenotype. In this case, the phenotype is the purple. And it's not until you have both copies broken, where you have both recessive alleles represented by these lowercase letters, that you actually show that recessive phenotype, in which case this would be having no color. Because I have no genes that can make purple pigment, now all I have is a white color that's left behind because I can't make that purple pigment. Okay, so that is the basics of genetics. It's what you need, that's the vocabulary that you need to know before we move on to the monohybrid crosses.